Good evening, students. In this video, today we are going to look at the structure of DNA and RNA, even the chemical components of it, where it is present, and how are they formed, how are they replicated. So let's begin. So here, as you can see, this is a cell. Okay, this is the picture of a cell, illustration of a cell, where you can see there is a lot of organelles present. This is a picture, okay, and there is a different organs present in the cell, and this is the nucleus. And here the nucleus is zoomed in to see the different parts of the nucleus. As you can see, they have mentioned the ribosomes, um, and inside the nucleus, inside the very core of the nucleus, there is the chromatin, DNA, and the nucleolus, nuclear lamina, inner nuclear membrane, outer membrane, and the nuclear pores. So there are more many nuclear pores through which many transfers happen outside and inside of the nucleus, and inside of in the core of the nucleus, the DNA and the chromatin substance are present. Now let's zoom in and see how what is the difference between the chromosome, DNA, and the gene, and what are the structures. So this is another picture of the nucleus. So you can see this is the nuclear envelope. This is a cell where the organelles are present. This is the nucleus and the nucleolus, and the DNA is present in the core of the nucleus. Now, so this is the chromosome. So let's, as I mentioned before, let's look at the difference between the chromosome, gene, and the or how is the structure of each. So as you can see here, this is a chromosome. Chromosome actually is a very coiled structure. It is in the coiled form inside the nucleus, and it takes the structure this with the two arms as in the chromosome when it is ready to replicate. And so it has a Q arm and the P arm. So the different parts of the chromosome, this is the Q arm and this is the P arm, and the both of the parts of the chromosome is linked in between by the centromere. It's called the centromere. Okay, and what is this chromosome made up of? Basically, this chromosome is of the coiled DNA. Okay, basically the DNA is coiled together to form this the chromosome. So when the DNA is coiled together and stacked upon each other, it forms the chromosome. So when we zoom into this DNA part, you can see a part of this DNA is called the gene. Okay, the part of this DNA is called the gene. This is one gene. This is a gene, and we can see that from this to a part here can also be called another part, gene. Okay, now let's look at zoom into the DNA. So actually when you zoom into this thread, when zoomed into, you can see that it takes this structure which we call the double helix model of the DNA. Okay, and why is it called the double helix model? Okay, we'll look into that. So as you can see here, the DNA is made up of many nucleotides. So if you rewind, we can, if you look at the uh, expansion of the name DNA, you can understand that DNA stands for deoxyribonucleic acid. What is the nucleic acid? Or this nucleic acid is nothing other than this nucleotide. What is nucleotide? Nucleotide consists of a sugar part, then it's linked to a phosphate. Then this phosphate, this sugar and this phosphate is linked to a base. Base means it is a nitrogen base. These are the chemical components of a DNA. When you say base, it can differ from this ATCG stands for adenine, thymine, cytosine and guanine, which we will get into in the next slide. So as you can see here, there is this bases are paired with each other. So there is two strands of this nucleotides which are bonded together using hydrogen bonds. So here the adenine and thymine has a double hydrogen bond, while cytosine and guanine has a triple bond. So here this sugar and phosphate forms the backbone of the DNA. Here as you can see, this strand of this DNA is basically this sugar and phosphate part. Okay, and the bases are bonded together to form a ladder-like structure, and this ladder-like structure is twisted together to form this DNA. Okay, it's when you untwist the DNA we get a ladder-like structure like this. Okay, and it is a sugar phosphate backbone. Okay, so as you can see here, this is a furthermore explanation from the next slide in this from the previous slide. So as you can see, as I explained in the previous slide, this cytosine is bonded with guanine, adenosine with thymine, and etc. So the cytosine only bonds with guanine, it won't bond together with thymine or adenosine. Similarly, adenosine will only bond together with the thymine. So this is the DNA ladder, and I was explaining before, this is the double helix. Now if we zoom into this DNA ladder, we can see that, as I was explaining before, this is the nucleotide. This small part is a nucleotide, and this is how each nucleotide is chaining with or is forming bond with the next nucleotide. This is how the DNA stays intact. Each nucleotide combines with the next nucleotides to form what? The DNA. Okay, and the two strands, there is the two backbones of the DNA is combined through hydrogen bonds from the nitrogen pairs. So as you can see here, you can see what you can see is a pentagon, right? In this pentagon, all these corners stands for the carbon atoms. Okay, all of these are carbon atoms. So here, this carbon atoms binds with the phosphate of the next nucleotide. Not the next nucleotide, of the same nucleotide, and this phosphate is linked with the carbon atom of the next nucleotide. So this is more detailed structure. I added this picture to show you the chemical polarity of the DNA strand. So as you can see, this is the 3' end of what? The nucleotide, and here is the 5' end. So 3' end means when we give numbers for each carbon atoms, the carbon atom that stands here is the 3' end. See, this is the 3' end. So but in the next strand, this is the 3' end, and the 5' end comes here, that is with the phosphate. So when one strand goes from 3' to 5' end, the other strand starts from 3' to 5' in the other way. Okay, so here if this from 3' to 5' end, in the other end it's the opposite, that is the 5' to 3'. This slide here explains how a DNA is replicated. So as you can see, the chromosome, this is the chromosome, this is the DNA, the coiled DNA. You zoom into the coiled DNA, and this DNA is ready for the replication. So this is the original DNA that wants to replicate. So when the DNA is ready for replication, a replication forks come in that divide the DNA into two separate strands. And this original strands, the two separate strands of the DNA is act as the original template for the DNA, for the new DNA or the daughter DNA. Okay, so also note that the colors of this nitrogen bases stand for each nitrogen base. So when the original DNA is divided into two particular strands, the DNA polymerase comes into play and it forms the other strand that is needed for to make the new DNA strand. So the DNA polymerase brings in the nitrogen bases that is need to form the new DNA strand here. So it brings all the necessary chemicals and the needed nitrogen bases, sugar, phosphate, etc. It brings in, and as you can see, it brings it forms bond. The ones that come and forms bond here is the one, for example, the guanine only forms bond with cytosine and the thymine only forms with adenine. So when a template is provided, the DNA polymerase is perfectly capable of forming the DNA that is like very similar to or the exact one that was the in the original DNA. So the nitrogen bases that comes in to form the daughter DNA is also uh, according to the template DNA strand that we have here. Okay, so that the daughter DNA that is formed is the exact copy of the parent DNA.